So here in zoology, so you have a very important chapter, that is principle of inheritance and variation. In short, so this entire chapter can be labeled as genetics. So this genetics, it's a branch of biology that deals with heredity and variation. So genetics is a branch of biology that deals with heredity and variation. Heredity means what? Transmission of characters from parents to offsprings is what you call it as heredity. The difference between the parents and the offsprings of the same parents is what you call it as the variation. Okay. So here, from NEET point of view, you need to understand the loss. Okay, so Gregor Johann Mendel, he's the father of genetics. So when he was alive, his work was not recognized. So when he was dead, his work was being recognized and he's regarded as the father of genetics. So here he proposed different laws. That laws you have to be perfect. So the first is the law of dominance. The law of dominance. So I'm touching those topics which are very important from NEET this thing. So they'll be asking more of question from these topics. So the law of dominance suggests that, so like if you have genes, okay, so a dominant gene, so in an organism, so if it is in heterozygous condition, if you have a dominant gene and you have a recessive gene, capital T is the dominant gene, small t is the recessive gene. So this is what you call it as the heterozygous condition. When an organism is in heterozygous condition, the gene which is dominant, okay, so the character of that gene will be expressed. So this capital T small t is a heterozygous tall plant. Mendel did research in pea plants. So now in that, capital T is for tallness and small t is for dwarfness. So this characters is been the dominant gene gives the trait tallness and small t gives the trait dwarfness. So now if you have two genes, this is a dominant gene, capital T, it is a dominant gene. Whereas small t, recessive gene. So first of all, you should know what is a dominant gene and a recessive gene. Dominant gene is the gene which expresses itself in presence of alternative gene. So recessive gene is a gene which suppresses itself when dominant gene is present. So now, if both the genes are dominant, then the plant will be tall, but it will be homozygous tall. If both the genes are recessive, then the plant will be homozygous dwarf. So when you have a mixture of one dominant gene and recessive gene in the individual, then the plant will be heterozygous tall. So no, what is the meaning of this terminology, homozygous and heterozygous? Homozygous means if both the genes are same, then it is a homozygous condition, a cell or a zygote with similar type of the genes. It is called as the homozygous condition. If you have a cell or a zygote with dissimilar type of genes, So that is what you call it as the heterozygous condition. Now here also the plant is tall, even here the plant is tall, but here as you are having two different genes, one is the dominant gene capital T and another one is the recessive gene small t. So that's why the plant is tall but heterozygous tall. But here, even though this alternative recessive gene is present, which character is being expressed? Dominant character, which is tallness, is being expressed. Okay, so the plant is tall, even though the recessive gene small t is present. So that is what you call it as the law of dominance. The law of dominance suggests that if a dominant gene is present along with any of the alternative gene, the dominant gene will be expressed and the recessive gene will be suppressed. So this is what you call it as the law of dominance. So this is the first law which is being proposed by Mendel. So the next one, so here you have understood these terminologies: homozygous condition, heterozygous condition, dominant gene and recessive gene. And so once they had asked about locus. So now suppose this is a chromosome. Okay, so you will be having a region. And one more thing, so before to that you should understand two more terminologies: homologous chromosomes and heterologous chromosome. So now if the chromosomes are a pair of chromosomes who are similar in their size, shape and morphology, is what you call it as homologous chromosomes. The chromosome what I have drawn here are what? Homologous chromosomes. So they will give you the diagram of the chromosomes, and if they are similar in their size, shape, morphology, everything, then those chromosomes are called as homologous chromosome, and always the homologous chromosomes will make up a pair. Now this homologous chromosomes, on the homologous chromosome, a region where these alleles are found. So now here you have capital T, small t. Here you have small t, small t. This point or a region where you have the alleles. What is alleles? A pair of alternate characters a pair of alternate genes which controls a character in an organism is what you call it as alleles. Now these alleles are found on which place? They are found on a place called as locus. So now what is locus? It is a point or a region on homologous chromosomes where alleles are found. So now capital T, small t, small t, small t, which is found in this region are what? Are alleles. So what are these alleles? Alleles are a pair of alternate genes which is controlling a character in an organism. So now small t is an allele which is controlling a character, doffness. Capital T are the alleles which are controlling the character, tallness. So did you understand what is allele? So it is a pair of alternate genes which are controlling one character. And where are these alleles found? These alleles are found on a place called as locus. Singular is called locus, plural loci. So this you have to remember the terminologies in case of genetics, and you should know what is homologous and heterologous chromosomes. Homologous chromosomes are similar in their size, shape and morphology, and heterologous chromosomes, they are not similar, they are dissimilar in their size, shape and morphology. And you should also remember one more terminology with respect to this, very important: phenotype and genotype. Usually they will ask phenotypic ratio and genotypic ratio of a monohybrid cross and a dihybrid cross. So you should know what is phenotype and genotype. So here, so this is a tall plant. As I have said, the capital T, two dominant genes are present. This is a tall plant. Even here you have two genes, but one is dominant gene and another one is a recessive gene. But it is in heterozygous condition. Here, this is also tall plant, this is also tall plant. But what is the difference? One is in homozygous condition, another one is in heterozygous condition. So that is what do you call it as the phenotype and genotype. All what you can see when you see a plant, you cannot tell it is a homozygous tall plant or it is an heterozygous tall plant. Morphologically, what you can see, it is, what do you call it as, phenotype. And then genotype. What is phenotype? The external morphological characteristic features. The external characters of an organism which are visible, or it is what you call it as the phenotype. Genotype means genetic makeup of an organism is genotype. Now you have capital T, tall plant, capital T, small t, tall plant, but tall plant is phenotype. But capital T is one genotype, capital T, small t is one more form of genotype. So these two terminologies you should remember. So they will ask you the phenotypic and genotypic ratio. So now the second law is law of segregation.
Phenotype is the external characters or the visible characters of the organism. Genotype is the genetic makeup of the organism like DNA, RNA, etc.
Okay, so which one is visible? Phenotype is visible or genotype is visible?
Phenotype is visible.
Okay. So what is locus?
The locus is the point where we find the alternative pair of the alleles which control the specific characters of the organism.
Other people can also interact with me. Alleles. Can anyone define what is allele? Allele. Just now I have explained. So directly they will give you capital T, small t. So this is so and so character. It is producing tallness. So in this they will give you 3-4 letter combination. In that they will ask you to select which one is the allele for tallness, dwarfness. Okay, so what is this allele? What have you understood when I have explained you the term allele? Define allele. Anyone?
A pair of contrasting traits is known
Yes,
as
yes,
allele.
tell. A pair of alternate genes which is controlling a character. It is what do you call it as? Allele. Okay. So now what does this law of dominance says? What does the law of dominance says? When you have two genes, dominant gene and recessive gene, whose character is being expressed according to law of dominance?
Dominant gene.
Dominant gene, the character of the dominant gene or the trait of the dominant gene is expressed. And what happens to the recessive gene? It is
Dominant
present.
gene suppresses the characters of recessive gene.
It completely dominates. It will not allow the recessive gene to express itself. So now we will move on to the next law, which was being proposed by Mendel, the law of segregation. To understand this law of segregation, you need to know the monohybrid cross. So first of all, can anyone tell me what is a hybrid? What is hybrid? What is hybrid? So if you don't know what is hybrid, then explaining this complete cross, It makes no sense. You should know what is a hybrid. Tell me in your own words, what have you understood by the term hybrid? Okay. So now I'm taking a plant, male pea plant, tall plant who is female. I'm crossing with the one more tall plant, male. Male tall plant is been crossed with a female tall plant. Will you get a hybrid from this? Will you get a hybrid? Will you get a hybrid when I'm crossing the same plants, tall plant and tall plant male, tall plant female? When I'm crossing this, will you get a hybrid? When will you get a hybrid? Okay, hybrid is an individual obtained by crossing two different parents. Hybrid is an individual obtained by crossing two different parents. Here you will never get a hybrid. Tall plant, you will not get a hybrid. So if a female is a tall plant, then male should be dwarf. So when you are making a cross between two different parents, then what will you get? A hybrid. So how can you define a hybrid? Hybrid is a individual obtained by crossing two different parents. They should be different. If you are crossing a tall plant with a tall plant, you will never get a hybrid. So when will you get a hybrid? When you are crossing the parents differing in characters. They should be different. Only then you will get a hybrid. So now when you are crossing tall plant with a dwarf plant, then you are getting a hybrid. So now one more additional term they have added over here, monohybrid cross. Monohybrid cross means, so it is a cross made between the parents differing in, mono means what? One. Differing in one set of contrasting characters. So now if you are taking only one character, then it is a monohybrid cross. Tall plant crossed with the dwarf plant will give you a hybrid. And such a cross where you are involved, where you are taking just only one character, it's what do you call it as a monohybrid cross. Now when you are crossing a tall plant with a dwarf plant, it's a monohybrid cross. So here these are the parents, P1. So here in genetics, we will not completely write the full name, parent. So here you use the technical term P1. So they may give you all these terms, technical terms P1, P2, F1, F2. So while explaining, I will be telling you, you have to make a note of this. P1 means first parental generation. P2 means second parental generation. F1 means first filial generation. F2 means second filial generation. Now this is the first parental generation, tall plant versus dwarf plant. So now here, what will be the genotype? Capital T, capital T. Initially in the first parental generation, you are taking only the dominant parents, dominant as well as recessive parents, but they will be in the homozygous condition. In the parental generation, first parental generation, the parents will be in homozygous condition. So the tall plant will never be capital T, small t. You usually select the what do you call it as the pure line. So they will be having the same genes. Dwarf plant, tell me what will be the genotype? Tell me.
Small t, small t.
Very good. Small t, small t. So now here gametes of P1. So here you will get capital T, here small t. Always make sure that you encircle the gametes. So sometimes in NEET, no, so they will in the option part they will give you this gametes. So who will be similar? in option A, they will give you capital T. Option B also capital T, small t, small t. So here they will encircle. For this, they won't encircle. So here they'll ask you to remove the odd one out. Okay. So the gamete which is not been encircled, you have to remove that. Understood? So always there is a rule that you have to encircle the gametes. Parents should not be encircled, but the gametes should be encircled. So now if they together, they form capital T, small t. So now this is F1 generation. What did I tell you for F1 generation? Tell me. F1 generation means? What is F1 generation? What is the complete abbreviation of F1? They will never give you the full form, filial, parental. What is F? F indicates filial generation. F1 first filial generation. Now this is the F1 hybrid. So now this is the hybrid which is obtained by crossing two different parents. One is tall, another is dwarf. Now you got F1 hybrid. Now tell me the phenotype of the F1 hybrid. Quickly. What will be the phenotype of F1 hybrid? Whether it is tall or dwarf, you should tell me now.
Tall.
Tall. Good. It is a tall. It is homozygous tall or heterozygous tall?
Heterozygous tall.
Good. Heterozygous. Tall. Now in case of the parent, this is homozygous tall or heterozygous tall?
Homozygous.
Homozygous twin.
Homozygous tall. This is
Homozygous tall.
very good. Homozygous tall, homozygous dwarf. But in F1 hybrid, you got heterozygous tall. So now, we will go for selfing. Selfing of F1. Selfing of F1. So we will go for a selfing capital T small t cross of capital T small t. Selfing means same thing, you are going to repeat both the parents. So this will be the P2 generation. P2 means what?
Second parent.
P2 means what? Second parental generation. Now what are the possible gametes over here? From P2?
Capital T small t, capital T small t.
Very good. Capital T small t, capital T small t. So now we'll put it in the checkerboard. These gametes. This is your checkerboard. Capital T, small t, capital T, small t. While writing in the checkerboard, you can, even if you don't encircle, it's okay. But normally when you write, you have to encircle. Here, whether you encircle or you don't encircle, so they will, it's not, when it is in the checkerboard. So it's like they follow this. So when you put the gametes in the checkerboard, so it's like an option that you can encircle the gametes or you can skip that. But if you are writing it individually, for calculation part, you have to encircle. So now, gametes you have arranged in the checkerboard. So checkerboard is also called as what? One more name for checkerboard. Tell me.
Punnett square. Punnett square.
Very good. Punnett square. A person by the name Punnett, he discovered, he found out. So that's why, based on to honor him, they have given the name Punnett square. So now what are the possibilities over here? Capital T, capital T, capital T, small t, capital T, small t, small t, small t. So now this is the tall plant or dwarf plant?
Tall plant.
Good. This one?
Tall.
Tall.
Tall plant.
This one?
Tall plant.
This one?
Dwarf.
Dwarf.
Good. So now the monohybrid phenotypic ratio is how much?
1:2
Three
is to 1.
I'm asking about phenotype. What is phenotype?
3:1.
3:1.
Good. 3:1. Phenotype means what you can see. How many tall plants are there? Morphologically, what you can see? How many tall plants?
3.
1, 2, 3. 3 tall plants and how many dwarf plants? 1. So phenotype is what? Morphological, physical appearance of an organism. Morphologically, what you can see? 3 tall plants and 1 dwarf plant. So this will be the phenotypic ratio. Then the monohybrid genotypic ratio.
1:2:1.
Very good. 1:2:1. 1 capital T capital T, 2 capital T small t, and 1 small t small t. So the genotypic ratio is 1:2:1. This ratios you need to remember. Okay. And after completing this dihybrid cross, I will teach you a technique how to, by just knowing the monohybrid phenotypic ratio, how you can calculate the, what do you call it, as the monohybrid, dihybrid phenotypic ratio. If you know the ratio of this, you can calculate aram se. Just by simple multiplication, you can calculate the phenotypic ratio of a dihybrid cross. Okay. So, do anyone know about the what is the dihybrid phenotypic ratio?
9:3:3:1.
Good. You know that 9:3:3:1. Okay. So now this is 3:1, right? Monohybrid phenotypic ratio. So 3:1 multiplied by 3:1. 3 3s are how much? 3 1s are
3.
1 3s are
3.
1 1s are.
1.
1.
So what did you get? 9:3:3:1. What is 9:3:3:1?
The phenotypic ratio of dihybrid.
Very good. Dihybrid phenotypic ratio. If you know the phenotypic ratio of the monohybrid cross, just by simple multiplication, by using the simple trick, if they give you any problem of and if they give you this ratio, phenotypic ratio, you can aram se by just simple multiplication, you can calculate the phenotypic ratio of a dihybrid cross. So likewise, do you know the genotypic ratio of the this dihybrid cross? Yes or no? Just tell me. If you don't know, I will teach you. Even this is also simple multiplication. Just you have to multiply. If you know, tell me yes. If no, I will teach you.
No.
Don't know? Okay. Now, what is the genotypic ratio of monohybrid cross?
1:2:1.
1:2:1 multiplied by 1:2:1. So 1 1s are
1.
1. 1 2s are, so here this with this and again same number and then the third digit. Understood? Then second one you are going to take, you are multiplying with this, you are multiplying with the second digit and you are multiplying with the third digit. Likewise the third digit also. Did you understand?
Yes.
So 1 1s are 1,
1.
1 2s are 2,
2.
then again 1 1s are 1. Then again you are taking the second digit, 2 1s are 2, 2 2s are 4, 2 1s are 2.
2.
Then again you are going to take the third digit, 1 1s are 1, 1 2s are 2, again 1 1s are 1. So there is one more small this thing you can, okay, first you listen here. 12, 12, 4, 2, 1, 2, 1. Just add 2 plus 1, 3, 4, 5, 6, 6 plus 4, 10, 12,
12.
13, 14, 15, 16. There are totally, when you go for a dihybrid cross, totally 16 boxes you have, right?
Yes.
Yes. So this is the genotypic ratio of a dihybrid cross. So if you want to remember in still more easily, I will give you one number: 12, 24, 12, 12, 1. Okay? 12, 1 to 12, 24, 1 to 12, 12, 1. Is it easy?
Yes.
What is the digit you should remember? 12, 24, 12,
12, 12,
12, 1. 12, 24, 12, 12, 1. After multiplication also you got the same answer. 12, 12, 24, 1 to 12, 12, 1. Understood? So if they give you any phenotypic and genotypic ratio of a monohybrid cross, through the simple multiplication, you are not going to waste the time and you are going to get the dihybrid phenotypic and genotypic ratio. Did you understand how you have to multiply? Okay, after me completing the class, you can just practice, and then if you are not understanding, you can clarify in my next class. You have to practice, or else in the digit. So if you are leaving just one digit also, you will not get the correct ratio. You have to multiply first digit with all the 3 digits, second digit with all the 3 digits, and again the third digit with all the 3 digits. Understood?
Yes.
Yes, ma'am.
Okay. Now I was telling you about the law of segregation. Now this was the monohybrid cross what we did, and we got the ratio 3:1 and 1:2:1. Yes, based on this monohybrid cross, so they put forward a law. What is that law? Law of segregation. So what does this law of segregation suggest? So when you are bringing the gametes, alternative gametes together in the F1 hybrid, okay, so when you are bringing the gametes, alternative gametes, so this is one gamete, this is one more gamete, others dissimilar gametes together in F1 hybrid, when they produce the gametes in the next generation, they get separated. So now here you can clearly see they are getting separated. So when the gametes are being produced in the P2 generation, they are not getting attached with each other. The gamete for capital T is separate, small t is separate. So they are getting separated. And there is no mixing or blending in the characteristic feature. So either you are getting a tall plant or either you are getting a dwarf plant. Did you get any plant with an intermediate character? Did you get any plant with an intermediate character? Did you get either tall or dwarf? May say, like, did you get neither tall nor dwarf? Did you get such character in the F2 generation? This is F2 generation. Did you get any intermediate characters over here apart from tall and dwarf? There were 3 tall plants and 1 dwarf plant. Did you get? Yes or no? So you didn't get. Why? Because the gametes are maintaining the purity. The gametes are segregating. What is the meaning of segregation? Segregation means separation. So they get separated. They don't get blend with each other. That's why either you are getting tall or dwarf. No intermediate characters. So that's why this is called as the law of segregation. This law of segregation, there is one more name called as law of purity of gametes. Law of purity of gametes. Why this is also called as law of purity of gametes? Because the gametes are maintaining their purity. How they are maintaining the purity? Without blending or mixing with each other and exchanging the characteristic feature, they are maintaining their self. That's why this law is also called as law of purity of gametes. Did you understand? So
Yes,
here,
ma'am.
ratio you should remember phenotypic ratio and genotypic ratio. Monohybrid phenotypic ratio is 3:1. Monohybrid genotypic ratio is 1:2:1. So this is very important. And if they change the plant and if they give you any ratio and they ask you to calculate the dihybrid phenotypic ratio, just take the value and multiply with the same. You will get the dihybrid phenotypic ratio and a dihybrid genotypic ratio. Did you understand? This calculation part, did you understand? Here in this chapter, a little bit
Yes.
of mathematic calculations will be there because they may ask you the problems. So genetic problems and the ratio you have to remember. So this you have to know. Instead of counting over there, you'll have very less time. So you have to remember this by using this code. What is this code? 12, 24, 12, 12, 1. You can remember easily. So multiplying takes time, but remembering in the form of this code is very easy. When you should go for multiplication, if they change the ratio of a genotypic ratio of a monohybrid cross and supposingly they instead of 1:2:1 they give 2:1:4. If they change the ratio and they give, so then this will not come. This answer will not come. You have to multiply and you should get the answer. For normal pea plants, so this is the answer. 12, 24, 12, 12, 1. Understood?
Yes.
So then the third law, the law of independent assortment. So the law of independent assortment states that when you're bringing many characters together, okay, so more than 2 characters, when you're bringing many characters together in F1 generation, the characters, they just get separated independent to each other. In law of segregation, you're just bringing only 2 characters. So, but in case of this, so to understand this law, you are going for a dihybrid cross. So what is a dihybrid cross? So it is a cross between the parents differing in 2 sets of contrasting characters. So when you understand, when we do this dihybrid cross, then you can understand the third law of Mendel, which is the law of independent assortment. So now what does this dihybrid cross? It is a cross between the parents differing in 2 sets of contrasting characters. Now you are taking 2 sets of contrasting characters. Round plant with yellow seeds. It's been crossed with wrinkled seeds, wrinkled and green seeds. So this is P1. So now what is the difference between a monohybrid cross and dihybrid cross? Tell me please. We did monohybrid cross first to understand the law of segregation. Now we are going for law of independent assortment. So for this, to understand this, we are performing this dihybrid cross. So what is the difference between the monohybrid and dihybrid cross? Tell me. How many characters am I taking here? And how many characters did I take in a monohybrid cross?
In monohybrid cross, we take only one character, and in dihybrid cross,
One
we
pair
take
of
2
character.
characters. Yes.
One pair of character.
Yes. ma'am.
One character means only tall or dwarf. Pair of character means tall, dwarf.
Yes. ma'am.
Okay, and you have to mention contrasting character. What is the meaning of contrasting character? Opposite character. Opposite of tall,
Dwarf.
opposite to round,
Wrinkle.
yellow, green. Understood? So there I took one pair of contrasting character. Here I'm taking two pairs of contrasting characters. So that's why it is a dihybrid cross. It is a cross made between the parents differing in two sets of contrasting characters. And a resultant hybrid which you obtain is a dihybrid. So I'll represent here by the letters capital R, capital R, capital Y cross of small r, small y. Capital R round, small r wrinkled. Dominant character, recessive character. Capital Y yellow dominant character, small y it is a green recessive character. So now in this character you should tell me once again which is the dominant and which is the recessive characters. Tell me which is dominant character.
Round and yellow is dominant, wrinkled and green is recessive.
Tell me in pairs. Contrasting character. Which are the contrasting characters over here?
Round
Tell me the
is
two pairs of round wrinkled is the first pair of contrasting character. Which is the second pair of contrasting character?
yellow and green.
Very good. So now this is the P1 parent. What are the possible gametes? Capital R, capital Y, small r, small y. So these are the gametes of P1. Now in case of F1, what will you get? Capital R, small r, capital Y, small y. So this is F1 generation. Tell me what is F1 generation, girls? What is the full form of F1 generation?
Filial generation.
Filial parental
Just
generation.
filial generation. I have given number also.
First
Is
filial
it just
parental
filial generation? Sorry?
First filial generation.
Okay, first filial generation. Okay, so now tell me the morphology of this first filial generation.
Round and yellow.
Very good. Round and yellow. Round and yellow. So why not wrinkled and green? Because you have small r also, small y also. Why not wrinkled and green?
But the capital R and capital Y are dominant.
Very good. Capital R represents for round, capital Y for yellow.
Yellow.
R is dominant over small r, that's why you are getting round. Capital Y is dominant over small y, that's why you are getting yellow. Now you tell me whether it is homozygous round and yellow or heterozygous round and yellow.
Heterozygous round and yellow.
Very good. Heterozygous round and yellow. So now we'll go for selfing. Selfing of F1. So for selfing of F1 means what will you cross? Capital R, capital Y, small y, cross of capital R, capital Y, small y. You are going to repeat the same thing. So now, what are the possible gametes for this? Tell me. This point, this is very important. They will give you this capital R, capital Y, small y, and they will ask you to write down the possible gametes from this hybrid. You now you should tell me how many possible gametes are there? How many possible gametes?
Four.
Put the arrow mark over here. I've already given the hint. Four possible gametes. Tell me the four possible gametes. Here you do the mistake. You should never select the same letter while selecting the gametes. They can give you in any combination. F1 hybrid like this, any combination. They can give you capital R, small y, small r, capital Y. They can give you any type of the parent and they will ask you to write down the possible gametes. Tell me what are the possible gametes over here? What are the possible gametes to this? I have already given you the hint. 4 possible gametes. What are the 4 possible gametes from this F1 hybrid? Tell me quickly. Okay, I will give you one more hint. One letter should be R, one letter should be Y, either capital or small. Now tell me.
Capital R, capital Y.
Good. Capital R, capital Y.
Capital R,
Then
small y.
very good. Then
Small. Capital Y, small r.
first you go with small r only. Small r,
Capital Y,
capital Y.
small r, small y.
Very good. So these are the four possible gametes. In a dihybrid cross, the possible gametes will be 4. In monohybrid cross, the possible gametes will be 2. See here, how many possible gametes were there? Here, 2 possible gametes. For monohybrid cross, if it is 2, then for dihybrid cross, it will be 4. So likewise, even for this, same. Okay. So now we'll put these gametes in the checkerboard. Capital R, capital Y, capital R, small r, capital Y, small r, small y. We'll put all these gametes in the checkerboard. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. I have written correctly? Yes? Have I done
Yes,
any mistake or I have correct?
Yes.
Then again in this direction, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So we have arranged all this in the checkerboard. Okay, now what will you get? Capital R, capital R, capital Y, capital Y, capital R, capital R, capital Y, small y. Any one of you tell me what will you get here? Each and everyone should answer me now.
Capital R, small r, capital
Capital
Y,
R,
small r, capital Y, capital Y. Capital
small r,
R, capital
uh,
R, small r, capital Y,
how
small y.
capital R, then
Small r,
small r, then
capital
capital
Y,
Y.
small y.
Next person.
Capital R, capital R,
Uh,
capital Y, small y.
good. Next.
Capital R,
Uh,
small y, small y.
next.
Capital R, small r,
Hmm.
capital Y, small y.
This
Capital R, small
all
r,
should all should interact with me, not just one or two.
capital R, small r,
Okay.
small y, small y.
Okay. Next. One by one. Yeah. सर फरहान सर आप ही सर
मैम
Yes. Yes, ma'am.
मैम कौन आंसर नहीं कर रही उनको थोड़ा ये मैं फुल स्क्रीन में रखे हूँ तो आई एम नॉट एबल टू सी कौन आंसर कर रही नहीं कर रही करके नाम
Okay.
खाली है यहाँ पे
जी we have some very bright students with us today. Anisa and Sheikh Aliza have always been answering, and Uzma is not answering, and Humeza is also not answering.
Uzma and Humeza.
Humeza.
Uzma should answer now. Uzma, tell me.
Uzma, can you please respond to your teacher, beta?
Uzma, what will I get here? What are the possible gametes who are here?
Are you following what the teacher is asking you, Uzma? No response. Okay. Anyone who knows the answer can answer, please.
Humaiza? Uzma and Humaiza, you have to interact.
Capital Y, capital Y.
Capital R. Who is this? Humaiza?
Small r,
Capital R, small r. Capital Y,
capital Y, capital Y.
Y. Okay.
Good, very
Then
good.
next.
Capital R, small r, capital Y, small y.
Very good. Next. Small r. Small r.
Small r, small r,
Capital Y.
Capital
Y.
Y.
Then small r, capital Y, small y.
Small r,
Next.
small y.
Capital R, capital Y, small y.
Small y.
Capital R,
Capital
small
R,
r,
small
small y,
y, small y.
small y. Likewise, small r, capital Y, small y. Small r, small y, small y. So this will be, tell me the phenotype. You got two capital R, two capital Y. What is
Round
the phenotype?
yellow.
Good. Round yellow. This one?
Round yellow.
Round yellow. This one? Round.
yellow.
Good. This one?
Round yellow. Round yellow.
Good. This one? Round.
Green. Round green.
Green.
Round yellow.
Round yellow. This one? Round green. So I'll put one mark over here so that you can clearly identify it when telling the phenotypic ratio. This one? Round
Yellow.
yellow. This one? Round yellow. This one? Very good. Wrinkled yellow. This one?
Wrinkled yellow. Wrinkled yellow.
Wrinkled yellow. This one?
Round yellow.
Round yellow. Round
Round green.
green. So one more
Wrinkled
round
yellow.
green eye. Wrinkled yellow.
Wrinkled green.
Wrinkled green. So totally, how many round yellow? How many? Totally 1, 2, 3, 4, 5, 6, 7, 8, 9. Round yellow, 9. Then round green. How many round green? 1, 2, 3. Yes? Then how many wrinkled yellow?
3.
Then how many wrinkled green?
1.
Good. So this the dihybrid phenotypic ratio is 9:3:1. And genotypic ratio you can either count like this or how many capital R, capital Y, capital Y. So you will, if you count like this, you will get the ratio 12, 24, 12, 1. So if you start counting, it will take a lot of time. In it, you will not have so much of time to count all the genotypes. So you just remember this short trick to know this genotype: 12, 24, 12, 12, 1. Phenotypic ratio is 9:3:1. Phenotypic ratio 9:3:3:1. Round yellow 9, round green 3, wrinkled yellow 3, wrinkled green 1. So now based on this dihybrid cross, what did they conclude? They concluded, so a law was been proposed, the law of independent assortment. What does this law of independent assortment say? When you bring many characters together, so here you are getting how many characters? 2 pairs of characters. How many pairs? 2 pairs of characters. Yes. So totally 4 characters. 4 characters are being together over here in the F1 generation. But when they produce gametes in the next generation, all of them, they separate independent to each other. They're getting separated or they're getting assorted. What is the meaning of assortment? Assortment means separation, assorting, separating. When they produce gametes in the F1 generation, they are getting assorted, they are getting separated. So this is what you call it as the law of independent assortment. And one more thing you should understand, the production of one character is independent of production of one more character. Supposingly, round is independent of yellow. Round is not dependent on green or yellow color. If round color was round, the shape was dependent on yellow, then round is coming with yellow and round is also coming along with the green color. Isn't it? Yes or no?
Yes.
Are you understanding? So round is independent, round shape is independent of color of the seed. Round, the expression of the round character is independent of color of the seed. Round can come with yellow, round can come with green. Similarly, wrinkled can come with yellow, wrinkled can also come with green. Likewise, you go for yellow color can come with round, yellow color can also come with wrinkled. Understood? This green color green can come with round, green can also come with wrinkled. Did you understand my point? The expression of the characters, the production of the characters are independent to each other. They are not dependent on each other. Even though you are getting 4 characters together, 2 pairs, that means 4 characters you are getting together in F1 hybrid, but still when they produce gametes, they are independent to each other. The production of the characters is independent to each other. Round is independent of yellow, round is independent of green, wrinkled is independent of yellow, and wrinkled is independent of green. Vice versa. Yellow is independent of round and wrinkled, and green is independent of round and wrinkled. So this is what you call it as the law of independent assortment. Did you understand?
Yes, ma'am.
That ratio calculation, did you understand? Just by multiplication, you can get the dihybrid from monohybrid ratio.
Yes, ma'am.
Okay, just you practice what I have taught you, and you just go through this chapter, and then again further we'll clarify. So better you just, if you interact more, then it will be a more interesting session.
Girls, all you people who are participants today, all of you really answered well. I'm so very happy to know that you people really especially Anisa, Sheikh Aliza, and Humeza. You people did very, very well today. I'm very happy that you are part of these sessions. Please continue to be so and try to make it more interactive next time.